Mom, where are you going?
Little Kettle, Mom is getting cloned.
What is cloning, Mom?
Cloning is the process of taking genetic information from one living thing and creating identical copies of it. The copy material is called a clone. History of cloning. In 1885, the German scientist Hans Adolf Idridis started researching reproduction with the first study of cloning. He demonstrated that the two cell sea urchin zygote could be made into two separate zygotes by shaking. In 1902, Hans Beeman divided a salamander embryo into two by using baby hair and demonstrated that early embryo cells retain all the genetic information required to build a new organism. This experiment is the first artificial induction to generate identical twins, triplets, and so on for natural cloning. Dolly the sheep was the first successful mammal to be cloned by Ian Wilmot and Keith Campbell in 1996 by a somatic cell nuclear transfer. Many more animal clones has been born since Dolly, and the technique is becoming more mainstream. There are a few types of cloning, which are therapeutic cloning, reproductive cloning, and gene cloning. Therapeutic cloning, which is known as somatic cell nuclear transfer (SCNT), withholds the most potential as medical advancement. Therapeutic cloning is the process where a 6% DNA is extracted and used to grow an embryonic clone. Instead of inserting this embryo into a surrogate mother, this embryonic clone is used to grow stem cells, and these stem cells could become the basis for customized replacements.
DNA cloning is the process of making multiple identical copies of a particular fragment of DNA. In a typical DNA cloning procedure, the gene or other DNA fragment of interest is first inserted into a circular piece of DNA called a plasmid. The insertion is done using enzymes that cut and paste DNA, and it produces a molecule of recombinant DNA. Next, the recombinant plasmid is introduced into the bacteria. The bacteria carrying the plasmid are then selected and grown up. As they reproduce, they replicate the plasmid and pass it on to their offsprings, making copies of the DNA it contains. Next, we will be discussing the significance of cloning, as cloning can be seen as important in making stem cells. Stem cells are used to build, maintain, and repair the body throughout an individual's life. As these processes are naturally occurring, they can be manipulated to repair damaged or diseased organs and tissues. Furthermore, a person is able to find out if he or she has inherited a gene on the chromosome from an affected parent by a procedure called genetic screening.
Other than that, cloning also plays a vital role in the agriculture and fishing industry by improving the production of livestock and crops with breeding stocks, as it is a more time-effective way to breed livestock. Besides, cloning can also make plants resistant to herbicides, pest damage, infections, and disease by improving the quality of crops. In fact, cloned plants such as wheat, rice, maize, soybeans, and potatoes have already been produced and ready to be introduced into agriculture. Now let's move on to the application of therapeutic cloning. Therapeutic cloning assists in obtaining organs for transplantation and treating injured nerve cells. According to the Health Resource and Service Administration, nearly 100,000 patients are waiting for a donor transplant, but only 20,000 patients perform organ transplantation. Thus, therapeutic cloning can ease the shortage of donor organs available for transplantation using cloned organs such as kidney, lung, liver, and heart to replace therapy and medication. Nevertheless, a new study shows that therapeutic cloning can treat Parkinson's disease in a mouse model. The scientists used skin cells from the tail of the animal to generate and customize autologous dopamine neurons, the missing neurons in Parkinson's disease. The mice that received neurons derived from individually matched stem cell lines exhibited neurological improvement. Gene cloning is essential in the synthesis of vitamins, hormones, and antibiotics. The gene prop developed from gene cloning has application in treating genetic disorder, cystic fibrosis. When cystic fibrosis transmembrane regulator (CFTR) gene for making CFTR protein does not work properly, the body produces thick, sticky mucus and very salty sweat mainly at the lung and pancreas. Gene cloning can produce a new correct sequence of the CFTR gene that will be placed into the cells of the human body, giving the cells the ability to function normal CFTR proteins. Moreover, gene cloning could also remove the defective gene and insert new and healthy gene into its DNA sequence is known as gene therapy that can treat deathly Alzheimer's disease, leukemia, and sickle cell anemia. The sickle cell anemia, which does not carry enough amount of oxygen throughout the body, can be knocking down the expression of the BCL11A gene to modify fetal hemoglobin and increasing the fetal hemoglobin, which does not sickle and directly reduce hemoglobin. Furthermore, gene cloning also shows contribution in agriculture, such as cloning in bacteria facilitates nitrogen fixation in plants. The nitrogen in the air was fixed for plant to use. Nitrogen-fixing bacteria along with the desired genes are used to increase the crop productivity and health while decreasing the use of fertilizer, and hence agrochemical-free crops are produced which contain higher nutritional value toward humans.
So now, let us talk about what are some advantages and disadvantages of therapeutic and gene cloning to mankind. Let's talk about the advantages first. Therapeutic cloning can overcome the high demand for organ transplantation. Since the cloned organs would have the same genes, the chances of rejection are nullified. Next, gene cloning can be used in curing incurable diseases, for example, cancer. With the advancement in gene therapy and gene cloning, we could then replace the genes which causes cancer. Now, let us go into the disadvantages. As we all know, therapeutic cloning requires many egg cells. But this process of obtaining egg cells from a woman's body is extremely painful. Secondly, there could be a possibility of overpopulation, because more and more humans will be able to survive diseases, including the incurable ones. Let's talk about the future of cloning. Therapeutic cloning would definitely be the greatest eventual benefit of the advancement in cloning. What's left is to improve the success rate of the individual steps in therapeutic cloning. Therapeutic cloning has a higher chance of success because of the lower rejection and the minimal ethical arguments from the society. Gene cloning, on the other hand, is already widely used, especially in gene engineering to produce insulin and insect resistance. With more research, gene therapy would be more advanced and more complicated diseases like cancer can be overcome. Let's go into the ethical issues in cloning. Many oppose harvesting stem cells from embryos in the blastocyst stage for therapeutic cloning, despite it being donated or produced in the lab, because many believe that life has already begun in the embryo. For reproductive cloning and gene therapy, many believe that the natural evolution process will be destroyed. They believe that we shouldn't play God and control the evolution process by reproducing human clones and edit the genes of our future generation. As for performing genetic engineering to produce fitter and more productive farmed animals, ethical issues that arise include physical pain and mentally distress of the animal used in the experiments. Though there are steps taken to minimize the pain and distress of the animals, there are evidence of public concerns that shows that the animals are still suffering. Let's talk about social responsibility. Cloning may save many lives, but the social issues that have arisen from within is unavoidable.
Cloned children would unavoidably be raised in the shadow of the donor, in such a way where its psychological and social development would be highly constrained. In terms of parental, the clones will have no biological fathers because the eggs are taken only from the mother. There are concerns that if a cell used for cloning contains mutation, the resulting clone may risk being born with aging and age-linked diseases. Cloned animals tend to have difficulties delivering live young and will develop abnormalities such as lameness. This could cause them to be treated with an overdose amount of hormones and antibiotics, which could bring harm to the human body when consumed. Ian Wilmot, the lead scientist responsible for creating Dolly, has warned that even slight imbalances in a clone's hormone, protein or fat levels can compromise the safety of its milk or meat.
To summarize, cloning shows contribution in both medical treatment and scientific research. However, we believe that cloning technology is not always fruitful, as it also provides negative outcomes such as varies in longevity, over-reliance on cloning technology, stem cells extraction from embryos for curing devastated disease, against the law of nature by creating clone babies, and so on.
Papa, it's time to clone.